In this lecture, we will learn about electron transport chain and oxidative phosphorylation. The free energy of electrons transfer from NADPH and FADH2 to oxygen via protein-bound redox centers and are coupled to ATP synthesis. In a nutshell, these complexes help in the lateral transfer of electrons across the inner mitochondrial membrane, thereby releasing protons into the intermembrane space, which further helps in the formation of ATP. Now we will discuss the detailing of each complex. The standard free energy required to synthesize 1 mole of ATP from ADP and PI is 30.5 kilojoules. Oxidation of 1 NADH results in the synthesis of 2.5 ATP and that of 1 FADH2 releases 1.5 ATP. Electrons are transferred through a series of 4 protein complexes through which electrons pass from lower to higher standard reduction potentials. That is, the complex 1 will be having the least redox potential when compared to complex 5, having the highest redox potential. Electrons are carried from complexes 1 and 2 to complex 3 by coenzyme Q or ubiquinone, and then to complex 4 by peripheral membrane protein called cytochrome C. The larger overall free energy change is broken into 3 smaller packets, each of which is coupled with ATP synthesis in a process called oxidative phosphorylation. The oxidation of NADH FADH2 is associated with the synthesis of about 2.5 and 1.5 ATP respectively. This stoichiometry is called the P/O ratio, or the ratio of ATP synthesized to oxygen atoms reduced. The 4 respiratory complexes of electron transport chain consist of several protein components that are associated with a variety of redox active prosthetic groups with increasing reduction potentials. The complexes are all laterally mobile within the inner mitochondrial membrane. Coming to complex 1 is called the NADH dehydrogenase complex, which passes electrons from NADH to ubiquinone, contains 1 molecule of FMN, 6 to 7 FES clusters that participate in the electron transport process. The FES clusters consist of iron and sulfide ions and are both coordinated to protein cysteine sulfidyl groups. The standard reduction potential of a given type of iron-sulfur cluster depends on its interaction with its associated protein as well as on its oxidation state. The coenzymes of complex 1 are FMN and ubiquinone are capable of accepting and donating either 1 or 2 electrons because their semiquinone forms are stable. These coenzymes provide an electron contact between the 2 electron donor NADH and the 1 electron acceptor cytochrome. Coenzyme Q's hydrophobic tail is soluble in inner mitochondrial membrane's lipid bilayer. In mammals, this tail consists of 10 C5 isoprenoid units and hence the name Q10. Coming to complex 2, succinate dehydrogenase complex. It passes electrons from succinate of citric acid cycle to coenzyme Q. With the participation of a covalently bound FAD, a 2 FES cluster, 4 FES 4S cluster, 3 Fe 4S cluster and 1 cytochrome B560. The standard redox potential for electron transfer from succinate to coenzyme Q is insufficient to provide the free energy necessary to drive ATP synthesis. Now coming to complex 3. Complex 3 passes electrons from reduced coenzyme Q to cytochrome C. It consists of 4 redox cofactors, 2 B-type hemes, Bl and BH, and a C-type heme C1, and two FES clusters. Cytochromes are redox active proteins which contain heme groups that alter between Fe2 plus and Fe3 plus oxidation states during electron transport. Each type of cytochrome contains differently substituted porphyrin rings coordinated with the redox active iron atom. A B-type cytochrome contains protoporphyrin 9, which also occurs in hemoglobin and myoglobin. In cytochromes A and B, both ligands are histidine residues, whereas in cytochrome C, one of its ligands is histidine and the other is methionine. BC1 complex consists of 3 common subunits: cytochrome B, which has Bl or B566, and BH or B562, and cytochrome C1 and iron sulfur clusters, 2Fe2S cluster. Complex 3 pumps protons from the matrix to the intermembrane space via the Q cycle, a type of redox loop. Coenzyme QH2, a 2-electron carrier, reduces 2 molecules of cytochrome and 1 electron carrier while transporting 4 protons. Both NADH dehydrogenase complex or complex 1 and complex 3 pump 4 protons each into the intermembrane space. Now coming to cytochrome C, it is a peripheral membrane protein that is loosely bound to the outer surface of the inner mitochondrial membrane. It alternatively binds with complex 3 and complex 4, thereby shuttles electrons between them. Cytochrome C's binding site contains several invariant lysine residues. 2 lysine residues, lysine 86 and lysine 79 of cytochrome C, interact with the glutamate 235 and alanine 164 of cytochrome C1 of complex 3. Reduced hemes are highly reactive entities transferring electrons over distances of 10 to 20 angstroms. Electron transfer occurs mainly through bonds rather than through space via quantum mechanical processes known as electron tunneling, largely mediated by van der Waals interactions and water-bridged hydrogen bonds. Now coming to complex 4, cytochrome C oxidase, also known as COX, catalyzes the 1 electron oxidations of 4 consecutive reduced cytochromes. And the concomitant 4 electron reduction of 1 oxygen molecule to yield water. Eukaryotic complex 4 is a dimeric transmembrane protein composed of 8 to 13 subunits. Among them, subunit 1, 2 and 3 are subunits 1 and 2 contain all four of its redox active centers, 2 A-type hemes A and A3, and 2 copper containing centers, copper A and copper B. Electrons are passed from cytochrome C to copper A, then to heme A, and then to heme A3. from there to copper B. Oxygen binds to A3 cytochrome or heme and is reduced to water in complex 4. Copper A contains two cysteine sulfur atom ligands and have two additional protein ligands, each of which shows an arrangement like 2Fe2S cluster. The two copper atoms are present in their +1 oxidation state. Here in complex 4, protons are transported by proton pump mechanism, as the redox centers need not be H plus carriers. In proton pump mechanism, conformational changes to the complex occurs as electrons are transferred and protons move in one direction. Reduction of oxygen by cytochrome C oxidase. The reduction of oxygen to 2H2O by cytochrome C oxidase takes place on the cytochrome A3 copper B binuclear complex. The fully reduced A3 is in +2 oxidation state and copper B is in +1 oxidation state. The fully reduced A3 copper B binuclear complex can readily contribute 3 electrons to its bound oxygen in reaching its fully oxidized A3. A total of 10 protons are collected into the intermembrane space from complex 1, 2, 3 and 4. Thus collected 10 protons are pumped into the matrix through ATP synthase or complex 5. We will discuss, this is also known as oxidative phosphorylation. We will discuss oxidative phosphorylation in the next lecture. Thank you.